All right, so what we were just talking about. Remember that with the intracellular receptors, we're talking about the lipid-soluble hormones. Which are also called bound hormones. And then with the membrane bound, we're talking about water-soluble hormones. Okay, so let's start with talking about the membrane bound receptors. All right, you've got two types of these. The G proteins, which you've seen from the special senses chapters. The G protein coupled receptors are the ones that have alpha, beta, and gamma subunits that go into the cell and do something. The others are actually receptors that directly alter the activity of intracellular enzymes. So you'll see exactly what that looks like in just a minute. So let's start with the G proteins. With G proteins, you basically have four options, four things that can happen when you bind a hormone to a G protein. One, the activated alpha subunit can alter the activity of molecules inside the cell, producing a cellular response. So this gets bound to, alpha goes into the cell, and it alters the activity of molecules inside the cell. Okay? So just like with anything else that has a ligand bind, when this lets go, these guys come back, whatever's happening stops. Okay? Now, the second result. An activated alpha subunit can combine with ion channels, causing them to open or close, depending on which one you're talking about. Some ions or molecules can act as intracellular modulators. Remember, intracellular means inside, modulate means to control. They enter or synthesize within the cell that regulate the enzyme activity of the cell. So if you look here, here's my G protein coupled receptor, there's alpha, here's a channel. Notice that alpha isn't bound to it, the channel is closed. But as alpha floats over and actually binds, all of a sudden this channel opens, and now calcium can come into my cell. Okay, so literally alpha is the garage door opener for this channel. When it gets activated in this particular pathway, now I open the channel, but it's opened internally. This isn't like with acetylcholine binding on the outside of the cell. That binding that we're talking about is happening inside. Now, when does it close? When alpha isn't there. When alpha isn't bound to it, it closes. So that's result 2. Result 3. Activated alpha can alter the activity of enzymes inside the cell. So remember I told you that it's kind of like an on-off switch or having the ignition key for a car. Well, in this case, G protein gets bound. Alpha actually binds to an enzyme in the membrane. That binding turns this on. So now that enzyme becomes active. In this case, it's adenylate cyclase. So it's taking ATP and turning it into cyclic AMP that can do something inside the cell. Is that important to know? No. What's important to understand is in some pathways, alpha is going to turn on or off enzymes and control their activity. So in some cases, it makes them work. In some cases, it turns them off. It just kind of depends on the circumstance. The difference is I'm not opening a channel. I'm not changing something inside the cell. I'm changing an enzyme and how it works. Now, the fourth result. It can actually alter the concentration of intracellular mediators other than calcium and cGMP. So it can actually cause the creation of certain molecules that control things inside the cell. Okay, so those are the G protein options. You have 4 options. These are all water-soluble hormones. How do I know? Because all of their binding sites are outside of the cell. Now, let's talk about, let's see. So all this is saying is alpha's working when the G protein's bound to. When the hormone lets go, alpha stops working. That's literally all this is saying, okay? If the hormone goes away, gamma, beta, and alpha come back to their G protein home. So that was receptors that activate G proteins. Now let's talk about receptors that directly alter the activity of intracellular enzymes. In other words, gamma, beta, and alpha have nothing to do with it. And again, these are membrane-bound receptors, which means these are water-soluble hormones. We're still there. We haven't changed what we're talking about yet. Okay, these receptors, when bound, directly alter the activity of intracellular enzymes with no second messenger needed. So if you look at this guy here, there's the hormone binding. This guy actually has the enzyme interacting with him already. There is no alpha, beta, nope. So this is more a direct line of he gets bound to, and then the guanylate cyclase gets activated, okay? So receptors, when they're bound directly, alter the activity of that enzyme without the second messenger, like I said. The altered enzyme, in this example, this is a membrane-bound enzyme, but sometimes they're not. It just kind of depends. The mediator enzymes act as chemical signals that move from where they're altered to the cytoplasm, where they can activate processes that produce a cellular response. So the fact that this guy is working means that it's producing this, which is basically the catalyst to do several other things. Okay? Sorry, a couple other things. Another example, well, I guess not another example. So increase or decrease in the activity of intracellular mediator molecules is one way that this can work. It can result in phosphorylating intracellular proteins. It can work as activating processes that produce the response of the cell. But either way, this is a direct link. There are no minions involved here. Now, this is kind of an interesting thing as well. So in this example, this dude here is the enzyme. So when my water-soluble hormone comes in and binds, it actually activates this enzyme directly, and it starts phosphorylating things. It starts adding phosphate groups to things. Okay? So some are bound, and a portion of their receptor on the inner surface of the membrane acts as the phosphorylase enzyme. Phosphorylase meaning I put phosphate on things. And it phosphorylates several specific proteins. These can be part of a membrane-bound protein or not, and they can influence the activity of other enzymes in the cell. Maybe those phosphorylated bits are actually going and influencing other things in the cell. Excuse me. So something that's kind of interesting, receptors that directly alter the activity of intracellular enzymes. So there's something called the cascade effect, okay? Most hormones act exponentially, basically causing a pyramid effect. So you activate one receptor, that activates 3 separate things. Each one of those activates 3 separate things. Each one of those activates 3 separate things. So as this step-by-step progression happens, I'm basically amplifying the effect. Few mediator molecules activate several enzymes, and each enzyme activates several other enzymes to produce a response. This amplification system exists in which just a few molecules can actually control the activity of a lot of enzymes. Remember I said, as long as the affinity is there, you don't necessarily need a ton of hormone. Well, it's kind of the same thing. As long as I've got this amplification happening, I'm amplifying that reaction with each step, I'm going to get a big, huge reaction, even if I've only activated a few of these. All right, so that was the membrane-bound receptors with my water-soluble hormones. Now, intracellular receptors, lipid-soluble hormones. Okay? They're the only ones that can get inside the cell. Remember, intra means inside. So, lipid-soluble hormones can cross that plasma membrane into the cytoplasm or even the nucleus of a cell, bind to their receptor. Here's the receptor inside the cell. This is aldosterone. It literally just crosses over and then can bind directly to its receptor. If these guys were in here, they could just also travel there without any issue. So the receptor location for my intracellular receptors is either going to be the cytoplasm or it's going to be the nucleus, okay? So what do these do? What is the function of this hormone receptor complex? Meaning here's my receptor and the hormone is bound to it. Well, they can alter enzyme activity inside the cell. As a unit, they can go and alter how an enzyme is working, whether it's working or whether it stops working. But one of the kind of cool things is they can actually bind to DNA itself and they can transcribe it. They can make an mRNA that's then translated into a protein. So they themselves can go into the nucleus, cause the creation of an mRNA that is then taken out and made into a protein. Remember when I said that things take time, right? And I said that it's good that we've got the spares, kind of like our little savings account in the bloodstream, not streams. This is why that's so important. When you have this type of reaction over here, they're not instant. It usually takes several hours between the time that it gets the signal to release the hormone and the time that you actually see a response observed. Like you can actually calculate the response. You got to remember, 1, I've got to release the hormone. 2, it's got to get into the nucleus and bind to its receptor. 3, that has to then go and read the DNA. Now, DNA doesn't just get read and poof, it's ready to go. You actually have to process that messenger RNA. You've got to take out introns. You've got to add a poly A tail. You've got to add the starter sequence that I can't remember the name of right now. It literally does have to be altered. Then, once it's altered, it can go out and be read by the ribosomes over here. Now, this is another thing. That translation, that's the word I'm looking for. The translation that's done by the ribosomes, again, is not ultimately the final product either. That translation goes to the Golgi, and at the Golgi, it's folded in a specific way. It has to have parts taken out, maybe some things added in, but it has to be finalized. All of those steps, you add up all of those steps, that's time consuming. So that right there means that if I need something immediate, remember the whole, I've got 4 triangles and I have to have 4 triangles all at the same time. If I don't, my body doesn't like it, right? If this takes that much time, having all of those spares in the bloodstream is very beneficial. It is very beneficial to have those. So hormone influence is actually limited via the hormone receptor complex degradation. How fast your body breaks these things down determines how much of this you're going to make, because if you're breaking it down lickety split, you're going to make a little bit. If you're, breaking it down over the course of several days, then you'll probably have quite a bit of this protein product that you've made because it's stuck around for a longer period of time. Again, that's half-life. If the half-life is really short, you're not going to have much product. If the half-life is really long, you'll have it for quite a long time. And if you really want all the details of what this figure is about, you can read that there. I'm not going into the details because I've told you what I want you to know. And that is the end of chapter 17.